We're going to be talking about the cell membrane and transport of materials in Chapter 7. In the 9th edition of our biology book, here's a good way to break up those pages, and always remember to try out those questions in the back of each chapter. So let's review. We've already talked about phospholipids, a type of lipid. A special type, though. Remember, most lipids are nonpolar and do not mix well with water. Part of that's true for phospholipids. Remember, phospholipids are special because they have that special phosphate group on one end. That makes that end polar because phosphate groups are negatively charged. And if they're polar, it also makes that specific end hydrophilic, meaning it likes water. The other end with the fatty acid chains, remember one of them is saturated and the other is unsaturated. That's just like any other lipid, and it's nonpolar or non-charged, making the fatty acid chains here hydrophobic, or it's repelled by water. When you put them in water, phospholipids naturally come together in this design here, where we have the hydrophilic heads pointing outward, either to the cytoplasm or the extracellular fluid where there's a lot of water, and those ends are hydrophilic, so they don't care. And then we have the hydrophobic tails pointing inward. That way they can kind of get away from water in that aspect. They're hydrophobic, they don't like it. So we say phospholipids can be arranged as a bilayer or two layers, looking like this. Now, because of their arrangement and because of their polarity or non-polarity, different things can get in and out. It depends what they are. So this phospholipid bilayer can act as a cellular barrier, a border, to keep what's inside the cell separated from what's outside of the cell. Lipids can go through the bilayer quite easily, as we see here. Sugars, water, looks like they're stopped, salt, and then wastes. So it's impermeable to polar molecules here because of this central area with these hydrophobic tails. So that means we're going to need some help to get some of these molecules across the phospholipid bilayer. It's not very thick. It's only 8 nanometers thick. Our cell membrane separates a living cell from an aqueous environment. Aqueous meaning water. And we already know we're about 60 to 70% water. So our phospholipid bilayer can help control traffic in and out of the cell, some things getting in more easily, like other lipids, and some things not so easily. It all depends on whether these molecules are polar or not. And of course, size. So what can help us get things in and out of the cell through that phospholipid bilayer? Well, this is where proteins come in handy. So besides having phospholipids surround all of our cells, we have proteins embedded in the phospholipid bilayer. Take a look at this protein here in purple. You can recognize all those elaborate folds that give our proteins that 3D shape. And they anchor themselves into the phospholipid bilayer. Some of them go all the way through. Those are called transmembrane or integral proteins. And some don't. Some just sit maybe on the bottom inside here, only go part way through. But they can act as channels and transporters to help things get through the cell membrane, like an amino acid, water, and salts and ions, and wastes, like ammonia. So again, transmembrane proteins go all the way through the phospholipid bilayer, and they create semi-permeable channels. That means that it's very picky about what gets through. Specific proteins are going to allow specific things through them. Take a look at that beautiful protein there anchored into the phospholipid bilayer. All those folds that give it its 3D shape. Keep in mind that part of the protein that anchors to the inside would be nonpolar, but parts that are on the outside up here, like the purple areas, would be the polar regions, which is part of how it gets anchored into the bilayer in the first place. And it's those amino acids that make up proteins that determine the polarity areas of it, whether they're polar or not. So here's some pictures. This one's an interesting protein embedded into the phospholipid bilayer. It's called an aquaporin, and its name helps to give you a clue as to what it does. Aqua for water and porin for pore. These are proteins that allow water molecules to go in and out of cells. And they weren't discovered too long ago, actually. They function through a conformational change. This means the protein can actually change shape or conformation as molecules are traveling through. Over here we have a proton pump, like a channel in a photosynthetic bacteria. The H pluses going through. I can show you that. There we go. And there's many functions of membrane proteins. There's one acting as a transporter. Remember, this one's a transmembrane or integral protein. Same thing here, an enzyme who receives a certain molecule, causes a reaction to happen, and then releases the product, ready to do the reaction all over again. This is very common to have these enzymes embedded into the lipid bilayer, and even in places like the mitochondria and the chloroplasts, where you have folded membranes inside the organelles that contain these enzymes. A cell surface receptor, again receiving a molecule or what's called a ligand, meaning it's a specific molecule that fits into it, like a lock and a key, which then causes a conformational change usually to this protein, which then causes changes to happen inside the cell. A surface marker. This is a protein, this purple part here, and then these little hexagons, remember those symbolize glucose. So this is like a protein that has sugar on it. It's called a glycoprotein. This would be what like A and B blood cells would look like, whether they have the A marker or the B marker. And those markers then are what's called an antigen, because it can alert a cell whether that is actually supposed to be inside your body or if it's an invader. They can act as cell adhesion and stick to one another, like these two proteins are doing here, causing these two cells to stick together, which is important in the building of tissues and organs. And then attachment to the cytoskeleton below. Here we have a transmembrane or integral protein connected to what's called a peripheral protein below it, and then those connected to the microtubules of the cytoskeleton. So many different jobs that they do. Again, there's some classes here. The peripheral protein does not go all the way through the lipid bilayer, but integral proteins or transmembrane proteins, you can use either word, they do. They go all the way through. For the peripheral proteins, those generally are the surface markers, the antigens, and the transmembrane proteins are usually the transporters, like the channels and the pumps. So when we look at the whole of the lipid bilayer that surrounds cells, we see this fluid-like matrix. It's not static. It doesn't just stay the same. It's constantly changing. There's a lot of activity going on and a lot of individual parts playing their role. Because of this, it's given the nickname the fluid mosaic model. Here we can see our different transmembrane or integral proteins acting as channels and transporters, receptors. We see some more peripheral proteins who are acting like cell markers. We see some of these proteins have a sugar tail on them of glucose. We even see some of the lipids, the phospholipids, have a sugar tail on them as well. Any place we see these little hexagons sticking up, those are sugars or carbohydrates. These yellow things, those are cholesterol. We see the cholesterol also embedded in the fluid mosaic model. That helps to give it more flexibility. These little phospholipids, they don't stay in one place. They're also moving around and switching places. So lots of different activity going on here. Hence the fluid mosaic model. So those membrane carbohydrates that we've been looking at, whether they're sticking off of a protein or sticking off of a phospholipid, it helps with cell-to-cell recognition. They act as antigens so that our cells can tell whether that cell actually belongs there or doesn't. If it doesn't belong there, it can alert the immune system to launch an attack that an invader has entered your body. Now that we know the makeup of the phospholipid bilayer or the fluid mosaic model, let's talk about movement across the cell membrane or transport. This is so important because a cell needs materials. Things need to be delivered all the time, and it needs to get rid of materials as well. So one of the ways that this happens is called diffusion. And the nice thing about diffusion is that it just works because of the second law of thermodynamics. The universe tends towards disorder, or what's called entropy. This is the second law of thermodynamics. And basically, when we're talking diffusion, what this is really saying is that molecules tend to spread out from where they're more concentrated or more organized, and then they spread out to where there's less of them. So going towards disorder. I know it sounds kind of funny, but that's actually what's happening. And because our cell membranes allow things through, it can allow molecules to diffuse across. So here we have some molecules of dye diffusing across the cell membrane, water on both sides, and it'll continue to do that until it reaches what's called equilibrium, so that the amount or the concentration of the molecules are the same on each side. Now, it does not mean that after equilibrium that everything just stops. There will be a continual movement of molecules across this membrane. It's just that when equilibrium is reached, that the amount moving to the left will equal the amount moving to the right. Remember, diffusion is movement from high to low concentration. And simple diffusion basically means that this is just happening because of that second law of thermodynamics. It's not taking any extra energy away from you in order to do this. It's just naturally happening because of the laws of the universe, moving from high to low concentration. Let's see that one more time, just like we see in the picture here. Looks like grape Kool-Aid. When you put it into water, it spreads out. This is a form of passive transport, and again, passive transport means no extra energy is needed on your part. In fact, water also moves in the same way. Water molecules like to spread out from where there's more water molecules to where there's less water molecules. When water moves because of simple diffusion, this is called osmosis. Another type of diffusion still follows the second law of thermodynamics, but it happens through a protein who we can call a facilitator. This protein facilitates the diffusion because it allows the molecules to go through it. It acts as a channel. And this protein that's acting as a channel is not using any extra energy. It would be in the form of ATP. It's not using any ATP in order to do this. It's just allowing those molecules through. All right, so let's review. For simple diffusion, we have regular diffusion of molecules. We have the special diffusion of water molecules called osmosis. And then facilitated diffusion when molecules go through a facilitator or a protein. All three of these do not require ATP, therefore they are a form of passive transport. They all move from high to low, high concentration to low concentration. Now there's another type of transport. The opposite of passive transport is active transport. This is when molecules need to move against the concentration gradient. Therefore, they're going to need some energy to do this. They're going to go from low concentration to high concentration. This will usually require ATP to prime the pump, which is usually a protein that's embedded in a cell membrane. And ATP will activate that protein or activate that pump, and then the protein has to change shape in order to carry those molecules across. Therefore, that protein is nicknamed a pump. Whoa, there's our ATP right there. So let's take a look at this pump working here. This one's called antiport. If we watch the molecules, let's try that again. We see it's making use of moving molecules both into the cell and outside of the cell at the same time. And again, that's called antiport. That's needing energy in the form of ATP to cause this protein to change its conformation or change its shape in order to make that happen. The one over here, this one is called symport. Unfortunately, I don't have the animation working on that one, but in symport, we would be moving both the amino acid and the sodium molecules in the same direction from the extracellular fluid outside of the cell to the inside of the cell. Again, that's called symport. So antiport, you have molecules going both directions using the same pump. And in symport, you have molecules going the same direction using the same pump. So there's our review. So getting through a cell membrane. Types of passive transport are simple diffusion, going from high to low concentration gradient. This would usually be nonpolar or hydrophobic molecules that would be able to get across the cell membrane all on their own through that lipid bilayer. Why nonpolar and hydrophobic? Remember, that's because of those fatty acids that point inward in the lipid bilayer. They have the properties of being nonpolar and hydrophobic. So any molecules that are just like them, they allow through quite easily, like other lipids. In facilitated transport, we need that facilitator, probably because the molecule trying to get through cannot pass through that lipid bilayer, so it needs to go through a protein instead because it's a polar or a hydrophilic molecule, meaning it loves water, and it's charged just like water. So it's got to go through a protein channel it's a facilitator to help it get through. But remember, all of these top ones here have in common, they're all types of high to low transport, and they do not require ATP. However, active transport, we've got to go against the concentration gradient. We're going from low to high concentration. We're using a protein pump who's changing its conformation. It's embedded in the lipid bilayer, changing conformation, allowing things either into or out of the cell, or sometimes both at the same time. This requires ATP. There's our visual of those, going from high to low, whether it's just through the bilayer, through a protein and facilitated diffusion, or through a pump using ATP. The top two, passive transport, and the bottom one, active transport. And remember, osmosis, the movement of water, is just simple diffusion, but of water molecules, going from more water to less water. How about large molecules? Because those things have to get through as well. Well, that is possible also. Remember I mentioned that those phospholipids are not just static and staying in one place. Those things are always moving around, and because they're moving around, it makes it very easy that they can merge with other membranes, such as like a vesicle moving around in a cell can hit the outside lipid bilayer and merge with it, or indent inward and bring in food and then close off into a vesicle. These types of things can happen because of those wonderful properties of these phospholipids. So when you are bringing in molecules, that's called cellular eating. That's a form of what's called endocytosis. Look at the prefix. Endo means inside. Cyto means cell. So you're bringing these things into the cell. And there's two types. Whether you're eating big molecules, that would be then called phagocytosis. Phago actually means eating. So it means cell eating. And then cell drinking, when you're bringing in like big gulps of liquid. That's cellular drinking. It's called penocytosis. Peno actually means drinking. And both of those, again, are forms of bringing things in, whether it be large molecules or gulps of liquid molecules. Both of those, endocytosis. And then we can also get rid of things as well. That's called exocytosis. Exo means exit or to remove from the cell. So think about a one-celled creature like an amoeba. When an amoeba eats, it actually has to do phagocytosis. It has to surround food particles and bring them into itself. Or get rid of waste. It doesn't have kidneys like you have, or a large intestine with a rectum and an anus. It doesn't have those things because it's only single-celled, so it relies on these vesicles that it can fill with waste and then dump them out of the cell through exocytosis. That's how single-celled organisms move things that are big into and out of the cell. So let's take a closer look at how that works, how that might look. Here's a phagocytosis. Here's some food, whether that be some bacteria, some organic matter, another smaller protist, or something like that. This could be an amoeba here, or it could be something like a white blood cell in your own body. And when it encounters this food, the cell membrane can indent inward, and it can actually bring it in and form a vesicle around it. Remember, the phospholipids are always moving and can close off as soon as that comes in and these two edges touch. Then, once we've got what's called our food vacuole, it can fuse with a lysosome, which has digestive enzymes, and then digest that food. It would then be called the digestion vacuole. This is the process of phagocytosis. It's a type of endocytosis or cell eating. Penocytosis, here we have some big gulps of liquid molecules, liquid and other molecules dissolved in it. It's just going to take a big gulp of that and move that stuff inward into the cell. And when these 2 points touch here, those phospholipids will merge, again, because they're always moving around. And then we'll have a vesicle with these molecules dissolved inside that we wanted to bring into the cell. And then the last one, a very specific process, is called receptor-mediated endocytosis. This is when it's going to be very picky about what it's bringing in, and there's actual receptors outside here, receptor proteins on the outside of the lipid bilayer, that when you get enough of them connected to the molecule that we want, that they actually move inward and then close off, so that we're ensured that we actually have what we want. So it's said to be triggered by a molecular signal. So really the only thing different about this one up here, penocytosis, and the receptor-mediated endocytosis, is that the receptor one has actual protein receptors, and it doesn't start moving inward until enough of those receptors have been triggered. There's a lot of videos you can check out and see this actually in action if you'd like to see that. One of the neat ones is an amoeba, which is pretty large, but it's engulfing another type of protist called a paramecium. That's pretty cool to watch. So I highly recommend taking some time and pulling up the slides and then clicking on some of these links and watching it. Let's look at the special case of water. After all, life depends on water. This picture here, this is a paramecium. It's a type of protist. Looks like it's going to have some water action here in a little bit. So the movement of water across the cell membrane is called osmosis. It's a simple type of transport. It does not require any extra energy in the form of ATP. So when we do this, we say that water moves from a high concentration of water to a low concentration of water across the semi-permeable membrane. So think about this for a moment. In this tube, it's a kind of a U-shaped tube. The semi-permeable membrane is in the middle, and we've got water with some molecules in it. The water is the blue stuff. So take a look here. Where is there more water? It actually is on this side right here, on the left side. Over here there's less water, but more of the dissolved molecules. So we say the water is going to move to the right. There's more water over here, so it's going to move from more water to less water. Another way that you can say this is that water likes to move towards more solutes. Those are the substances dissolved in the solvent water. So water likes to move towards more solutes and dilute it out, and that's exactly what's going to happen. So if we let this proceed, after a few minutes, our water levels are going to change. And we're going to see the right side raise and the left side lower until equilibrium has been reached. So we have some vocabulary to keep track of this. The word hypertonic. This is when there's more solute present and less water. And it's pretty easy to remember because hyper, I think about like sugar, or something's really sugary, something's really salty. So that means more solutes. So in a hypertonic solution, you've got a lot of solutes present, like salt or sugar. A hypotonic means less solute or more water. Hypo always means less or below. And then iso means equal, that you would have an equal amount of water and equal amount of solutes. So if we look at what's happening here, this is the hypotonic side and the hypertonic side over here. So which way will the water move? It'll go towards the right, because water always moves across the semi-permeable membrane from more water to less water. Or, remember, you can also say it, water moves towards where there's more solutes. It's almost like water has a brain of its own and it's trying to dilute it out. So let's look at some cells and how this affects actual cells that have been put into solutions. So in this case, let's see, we've got a hypotonic solution. So this top one is an animal cell, and it's been put into a hypotonic solution. It's probably just pure water. And because water likes to move from more water to less, water is going to move into that cell because that cell has different solutes dissolved inside of it. So water moves inside, and then it ends up bursting, which means it lysed. Remember that prefix lyse, like lysol and lysosome? It means it broke it. But in a plant cell below, the same thing is happening, definitely. But a plant cell actually has that cell wall to give it extra support. So our plant cell won't actually lyse or break apart. It'll hold it, but there'll be an appropriate amount of pressure then to keep that plant cell looking nice and square or rectangular. When that happens, we say that cell is turgid or normal. It's normal to have that extra water in there and to have pressure against the cell wall to make it hold its position. In this next one, we have an isotonic solution, so that means the amount of solutes in the solution out here equal whatever is in the cell. That would have to be mathematically calculated or figured out ahead of time, or experimentally figured out. And so the amount of water moving into the cell and out of the cell is in equilibrium. For an animal cell, that would be what we want, because we don't have any extra cell wall to protect us. So that would be just a normal cell, like in our blood or in our cellular tissues. And we would not have cell lysis. It would not break up. In the plant cell below it, though, even though isotonic sounds really good for an animal cell, it's not quite as good for a plant cell. We say it's balanced, but it's kind of lacking some of that pressure that it had back over here in the hypotonic solution. And so we say this plant cell here is flaccid. It's just not quite as plump or filled up as our turgid cell over here. And then finally here, if we put our cells into a hypertonic solution where maybe it's really salty, or maybe the solution here is really sugary, much more than what was inside the cells, look what happens to our poor animal cell. Water likes to move from where there's more water to less, and in this case there was more water inside of our animal cell, and so it moved outward into the solution, therefore shrinking that poor cell. This would be a situation that we would not want to happen inside of our bodies, like when we get really dehydrated and our blood loses water through sweat and urination, and so then we have more solutes in our blood than normal and less water, so we lose water from our cells, and that can cause death. And then our plant cell, we see that's happening as well. Remember, there is a cell membrane in here, and what's happening is it is losing water just like the animal cell. The cell wall still holds up, but our membrane no longer holds up, and we see it also shrinking. This is called plasmolysis. We say these cells have plasmolysized and have lost water. So in the plant cell, we see the membrane shrinking away from the cell wall. So these are situations we would not want. This would happen when you don't water your plants. And again, when we get dehydrated. Same thing with animal cell in the hypotonic solution. If you were to sit down and drink 6 gallons of water or something like that, you would make your blood really watery. And this could happen to your red blood cells and other cells that blood comes into contact with. And, well, then you know what happens when red blood cells lyse or break apart, then death can result. So we continually manage our water balance. That's part of maintaining homeostasis for us. And it is, again, a tiny bit different here for the animal and the plant cell. A hypotonic solution would be like a cell that lives in fresh water, and like a freshwater amoeba or a freshwater paramecium. Both of those are protists. And they would have to constantly deal with water moving into them and constantly deal with the danger of cell lysis. So an amoeba and paramecia have little tricks up their sleeves. If they could have sleeves, they can actually pump water out. So here's a paramecium. A paramecium collects water in a vacuole, and then that vacuole can merge with the sides, and can dump it out. They continually do that when they live in fresh water like that. Water will continually go from more water to less water, meaning it will move into them towards more solutes, which they have in their cells. It's neat in this picture, you can actually see solutes and things in their cells. And so they continually have to pump water out. And amoeba also do this. Here you can see the water vacuole has shrunken and will then begin to fill up again over time. So that's called a contractile vacuole when that happens. Alright, in a hypertonic environment, remember, some organisms actually live like in the ocean where there's a lot of salt water and constantly have to deal with managing their water balance in a hypertonic environment. If we put our cells into a hypertonic environment, look what happens. They shrivel and shrink. So some organisms, like a shellfish, take up water or they pump out the salt to help deal with this. We can normally see plasmolysis in a plant cell when they wilt. But notice when you give them, if you give them water in time, they can recover. And in an isotonic environment, we have equilibrium. So somebody at one time sat down and they figured out that giving fluids to someone can help them out. But the fluids pumped into our bodies in the form of an IV have to be maintained at a certain solute level. It can't be just straight water. It has to be a certain concentration. So they figured out what that concentration was, and so that becomes an isotonic solution compared to our cells. And remember that when we reach equilibrium, when a cell reaches equilibrium with its environment, it's not that those molecules just stop. They're still moving across that cell membrane. It's just that what's going in is equaling what's going out. Here's the case of aquaporins. Water can actually move quite rapidly into and out of cells. And so there was evidence that there had to be something, because water wouldn't move that rapidly across that lipid bilayer because of the polarity. So there had to be some evidence. There's got to be something helping it out, which turned out to be these water channels that were discovered, and they were named aquaporins. So let's see, can you figure this out? You could pause the video and take a look here. We have a cell in a certain environment. This is a model, by the way. They're using a material called dialysis tubing that they have to fill and then tie off on one end. It's like a little balloon, but it's made out of a material called dialysis tubing that is like a semi-permeable membrane, allowing some things in and out at a certain rate. So the cell compared to the beaker, let's see, the cell is 0.03 molar sucrose and 0.02 molar glucose, so add that up to get the concentration. The environment is 0.01 of each of these three sugars, so add that up to get the environment concentration. And then let's compare. Is the cell compared to the beaker hypertonic or hypotonic? There it is. It's hypertonic because the cell has a higher concentration than the external environment that it's in. How about the beaker compared to the cell? Well, that would be the opposite. It's hypotonic compared to the inside of the cell. These words, hypertonic, hypotonic, you use them to compare two areas with one another. And often in biology, it's comparing a cell, what's inside versus what's outside. So which way is the water going to flow? If you need to pause the video to think that out, do that now. Otherwise, I'm going to tell you. Water is going to go in. Well, why is that? Well, remember, water moves from where there's more water to less water. So water is going to move from outside here, where it's hypotonic, to the inside, where it's hypertonic and there's more solutes. Remember, water likes to kind of act like it has a brain of its own and move to where it can dilute out all these solutes inside this cell. So what's going to happen to the shape of our cell? Well, if water moves in, it's going to grow. Here's an example of some onion cells. Here, salt water has hit this onion cell. We can see the cell wall holds up just fine, but the cell membrane, which this is a purple onion, so the cell membrane has purple dye or purple pigment on it, and the purple pigment, the cell membrane here, has shrunk inward. The salt water has not quite yet hit this one yet, but it will. Alright, so here's our learning targets for our chapter. I can explain the components and properties of a cell membrane. I can explain what's meant by the fluid mosaic model. Distinguish between the following pairs or sets of terms, like the difference between a peripheral and integral or transmembrane protein, a channel and carrier protein, diffusion, osmosis, facilitated diffusion, and active transport. So you're able to distinguish between all those terms. And the words hyper and hypo and isotonic, those types of solutions. Explain how transport proteins facilitate diffusion. We haven't got to this one yet, number 5, but we will look at a specific case of the sodium-potassium pump. Explain how large molecules are transported to the membrane, like, remember, endo and exocytosis, phagocytosis and pinocytosis. So define those types of movement. And we haven't got to number 8 yet. Diagram and explain water potential and calculate water potential. So we'll get to those when we do our labs in this chapter. All right. Let me know if you have any questions.